Hello everybody, my name is Jonavi and I will be talking about associative learning in Cnidaria. I'm going to start out by talking about basal phyla like Cnidarians and their nervous systems and comparing these to the nervous systems of Bilateria. Then I'll move on to defining and exploring the different types of learning present in Cnidaria. Of all animals, 4 phyla are considered to be basal, while the rest fall under the umbrella of Bilateria. Bilaterians have bilateral symmetry, like the name suggests, and they have concentrations of neurons that form a brain. In this image on the right, you can see the nervous system of a tardigrade in blue, including a centralized brain that you can see here. The 4 basal phyla are Ctenophora, Cnidaria, Placozoa, and Porifera. Of these phyla, 2 have no nervous system at all. These phyla are Porifera and Placozoa. There is no evidence of associative learning in these 2 phyla. The other 2 basal phyla, Ctenophora and Cnidaria, do have nervous systems, but these are in the form of nerve nets without a centralized brain, unlike in Bilaterians. A nerve net is a mesh-like nervous system where signals can be transmitted in any direction. You can see the nerve nets of Ctenophoras on the left here, and the nerve nets of Cnidarians on the right. Cnidarians form the closest group to Bilaterians on the animal evolutionary tree. Because of this, the learning capabilities of Cnidarians can provide insights into the origins of learning, cognition, and neurobiology in Bilaterians. It can also increase our understanding of the learning capabilities of the nervous system prior to the development of a brain. So let's talk about learning. The ability to form memories and learn allows animals to change their behaviors based on prior experiences. Learning can be broadly characterized into 2 groups, associative learning and non-associative learning. Associative learning occurs through the pairing of 2 previously unrelated stimuli. The best known example of associative learning is Pavlov's experiment with dogs, which is a form of classical conditioning. In this experiment, Pavlov conditioned dogs to salivate at the sound of a bell. He did this by pairing the natural response of salivation in response to food with the sound of a bell. Over time, dogs associated the sound of the bell with food, resulting in salivation at the sound even in the absence of food. In this case, food serves as the unconditioned stimulus, and salivation is the unconditioned response to the stimulus before conditioning. The sound of the bell is a neutral stimulus that initially elicits no response in the dog. After the conditioning period, the bell becomes a conditioned stimulus, and it elicits a conditioned response, which is salivation in this case, in the dogs. The other main form of associative learning is operant conditioning. In this form of learning, consequences are associated with a voluntary response, which is different from the involuntary responses in classical conditioning. Behaviors can be reinforced by giving rewards. An example of this is when you give a dog a treat to train them to do a trick. Another form of operant conditioning is giving punishments to decrease behaviors. Now let's talk about non-associative learning. The 2 main forms of non-associative learning are habituation and sensitization. Habituation is the decrease in response to a stimulus that is repeatedly presented. For example, if you gently touch a snail, it'll withdraw back into its shell. But if you repeat this enough, the snail will eventually stop withdrawing as it learns that the gentle touch is not a threat. On the other hand, sensitization is when repeated exposure to a stimulus increases the response to that stimulus. This can be done with repeated stimulus of the same kind or the presentation of some unrelated stimulus. An example of this is when a person becomes more on edge after hearing loud noises. Both habituation and sensitization have been repeatedly and strongly demonstrated both in and out of Bilateria. Even some bacteria and single-celled organisms appear to be capable of these forms of non-associative learning. They have been confidently demonstrated in Cnidaria as well. On the other hand, associative learning was previously thought to require a centralized brain, but some recent studies are bringing this into question. Studies showing classical conditioning in sea anemones have actually occurred as early as 1905, but these early studies lacked the full controls and reporting needed to consider their results definitive. But in 1975, a study of classical conditioning in sea anemones was done that included a full suite of controls. This study was replicated in 2023, which strengthened the evidence that associative learning could happen in Cnidaria. In both these studies, light was used as the neutral stimulus and paired with a shock as the unconditional stimulus. Let's take a closer look at how that works. In response to an electrical shock, sea anemones retracted. This reversible reaction is an unconditioned response to an unconditional stimulus, the shock. In response to light, anemones did not respond. That is because light is a neutral stimulus that does not elicit any response in anemones. Researchers paired the unconditioned shock stimulus with the neutral light stimulus repeatedly. The anemones retracted during these trials because of the shock. After conditioning, when researchers exposed anemones to light alone without any shock, they retracted. This shows successful classical conditioning, with the light becoming a conditioned stimulus and the retraction response becoming a conditioned response to that stimulus. This is a strong example of classical conditioning. The replicability and controls used in these experiments provide solid evidence that the response is in fact associative learning. While those previous studies all looked at learning in sea anemones, there has also been a study on another group of cnidarians, the box jellyfish. This study looks at operant conditioning, not classical conditioning. This study focused on the Caribbean box jellyfish, which has high behavior complexity and image-forming eyes, unlike anemones. This jellyfish is capable of visually guided navigation and can avoid obstacles. This jellyfish has a concentration of neurons in the eye-bearing sensory structures that allow for visual processing. These visual structures mediate a visually guided swimming behavior to avoid obstacles. The jellyfish uses the visual contrast between an obstacle and the surrounding water to stop at the correct distance from the obstacle. The Caribbean box jellyfish forages between prop roots in their mangrove habitat, which you can see in this image here. For this reason, it's important that the jellyfish can stop at the right time. If it stops too late, it'll crash into the roots, but if it stops early, it won't be able to fully explore its environment. This animal has a very fragile body, so it's very important for it to avoid damage through collision with obstacles in the environment. The operant conditioning experiment involved low-contrast obstacles that the jellyfish would initially collide with when they were first exposed to them. When the jellyfish were repeatedly exposed to the obstacles, they got better at avoiding them. The jellyfish learned to stay further away from the obstacles, and they would engage in obstacle avoidance behavior more often. Obstacle avoidance behavior is a rapid burst of movement that a jellyfish uses to turn itself around. These adaptations significantly decrease the jellyfish's collisions with the wall. This suggests operant conditioning because a negative consequence, collision with an obstacle, resulted in a change in the animal's behavior to avoid the consequence. In this experiment, the box jellyfish learned to associate the aversive stimulus of collision with the presented obstacle. Because of this, they avoided the obstacle as a result. This appears to be the first example of operant conditioning in Cnidaria, as well as the only strong study examining associative learning in jellyfish. These recent studies present strong evidence challenging the prior belief that a brain is needed for associative learning. Additionally, it's possible that a nervous system as a whole is not fully necessary. One study demonstrated associative learning in plants, but that study has failed to be replicated. Associative learning has also been demonstrated in some single-celled eukaryotes, but once again, the evidence for this is not very strong. These uncertainties do, however, show that more research on learning in animals without a brain is very much necessary to fully understand what structures are needed for associative learning and what organisms are and are not capable of this. As a basal phyla, Cnidaria holds a key position for understanding basic neuronal function. Associative learning has been demonstrated by studies showing replicable classical conditioning in sea anemones. The recent study in box jellyfish also shows the possibility of operant conditioning in Cnidaria, but this has yet to be replicated. These findings challenge the prior belief that associative learning needs a brain to occur. Further research is still necessary to determine what is actually necessary for this process. Thank you for listening.